Welcome to Backbenchers. I hope everything is going well with you all and I hope you all are fine. Today we will be continuing with the fifth portion of the chapter of absorption by roots under the playlist of plant physiology. We have already finished the first four parts of the chapter. So if you haven't seen them, then please go and check the videos out. I will definitely provide the link in the description box. Let us quickly have a look in the division of the chapter. The first was introduction part, which we have already finished. Second is roots and its advantages, which is also done. Third was osmosis and its type, which we have discussed already. And fourth was terms of osmosis and its significance and tonicity, which is also done. In today's session, we are going to be discussing about flaccidity, turgidity, and its significance. It is going to be a very small session as far as I expect, because this session does not contain a lot of things which is needed to be discussed, because already you all know these things. Sort of you all know. We are just going to be framing all of these definitions that you already know from the beginning, and then the session is going to be ended with the significance of turgidity. Then these are the rest of the portions of the chapter. We have come to an end of the chapter almost. We are left with two more sessions, and then I have kept a separate doubt class for you all, in which I'll be answering all of your doubts. So please, whatever doubts you're having, do not forget or do not hesitate to put them down in the comment section. I'll definitely answer them by the end of this chapter. Let us start with today's session. We have at first some questions from previous session. Let us have a look into them. First question is difference between osmotic pressure and osmotic potential. While teaching about osmotic pressure and osmotic potential in the previous session, I have discussed already the difference between them. There is only going to be one point of difference, a single point. So make sure you write that point correctly. Let me just write here, single point for you all, and do not forget. If you do not remember the concepts from the previous session, then you can go and watch the videos once again. There is no harm in that. And if you want, you can write the answers of these questions in the comment section. I'll check them out for you all. And the second question is a fill in the blank type question. It's a short question, MCQ types. Endosmosis takes place in a dash solution. While answering these kind of questions, please think twice while answering. Why? Because these questions are confusing and a lot of students make mistakes while answering this question. So make sure you give the answer second thought and then only go for answering this question. Now let us move on with today's session. The first thing that we are going to be learning in today's session is turgidity. The topic of turgidity we are going to be seeing first. And we are going to understand turgidity with the help of this beautiful and simple diagram that we have here. This diagram shows us a plant cell which is very stretched. You all can see it. The plant cell is pretty much stretched and it is completely filled with water. That is what this diagram is trying to tell us. So first of all, we can understand that this is the plant cell. How? Because we can see a cell wall here, and then inside the cell wall, this layer is the cell membrane. Let me just use another ink. This layer is the cell membrane, and let's mark it as the cell membrane. Okay, wait one second. So this is, if you all remember about the previous session in which we discussed about the three types of solutions, I want you all to just concentrate in the hypotonic solution. We will just be considering hypotonic solution for understanding turgidity. So focus on hypotonic solution. If you all remembered what happened in hypotonic solution, then water was entering into the cell due to a difference between the concentration of the surrounding and the cell. So water was entering inside the plant cell, and excess amount of water was making the plant cell to stretch, that is to become tighten. That condition is only known as turgidity. So turgidity is nothing different than hypotonic solution condition, but the only difference lies is there is no presence of solution in turgidity. Turgidity is just a normal state of a particular cell while absorbing substances or while in the process of absorption. So turgidity is not involving, does not involve, let me just write it here, does not involve any presence of hypotonic or any other solution. I'm just writing a hypotonic solution because we are putting focus on that more. So it doesn't involve any solution, but the rest of the things are same. Let us quickly have a look into the definition. It is the condition in which the cell can absorb no more water. One more new term is being added here. Not something which is totally new, because in the previous session only when we were discussing about hypotonic solution, you have already known about it. So it is a condition in which the cell can absorb no further amount of water. That is, the cell is completely filled with water. In this diagram, if you see, this is the nucleus and this is the vacuole. The vacuole should have been bigger. This should have been the size of the vacuole which I just drew. This should have been the actual size of the vacuole, because the vacuole is filled, completely filled with water, and it can absorb no further water. In this condition, what happens? The cell is more concentrated. Concentrated with what? Concentrated with water molecules. We are only concerned about water molecules here. So I'm just going to write it again. Concentrated with water molecules. That is what is our concern. Concentrated with water molecules, then its surrounding, hence it is stretched or enlarged. I'll highlight these two terms so that you never forget. Stretched or enlarged. Or enlarged under a pressure, a position which is known as turgid. Okay, so when there is excessive amount of water, the vacuole is going to enlarge in size, and when the vacuole is going to enlarge in size, the cell contents are going to go to the periphery of the cell. Periphery means at the sides of the cell, that is in the border areas. Let me just help you understand. So the vacuole has increased in size, and therefore what will happen? The cytoplasm of the cell is going to move to the corners of the cell, which is known as the periphery. The border which I just did, it is known as the periphery of the cell. Due to which a pressure is being exerted on the cytoplasm or on the other parts of the cells apart from the cell vacuole. Due to this pressure, a condition is developed in the cell, and that condition is known as turgid. That position or that situation of the cell is known as turgid, and the whole phenomenon is known as turgidity. So turgidity is a particular phenomenon in which the cell can absorb no more water, and it is stretched or enlarged under pressure, a position which is known as turgid. A position which is known as turgid. That is all about turgidity. Now, turgidity causes two types of pressure to act on the plant cell. In the previous slide only, I was talking about a pressure which is exerted by the cell vacuole. Now, not only the cell vacuole exerts a pressure, there is one more pressure which is exerted inside the cell or within the cell when the cell is in a turgid condition. What are those cells? What are those, what are those pressures? Wall pressure and turgor pressure. These two types of pressures are there. Let me just draw a plant cell for you all, and then let me just explain that how does wall pressure and turgor pressure happen. So suppose this is a wall pressure, this is the cell wall. and then this is the cell membrane inside the cell wall. This is the cell wall, and this is the cell membrane of the plant cell inside the cell wall. Now we are considering the cell to be present in a turgid condition. This is the large centrally located vacuole. Okay, let me just change the color of the ink here. A large centrally located vacuole which is containing sap. Then there are organelles. Then cytoplasm is going to be there. Organelles, for example, nucleus is going to be there. Mitochondria is going to be there. Ribosomes are going to be there. Just consider these to be the organelles, and this is the cytoplasm. Let me just matrix-like structure is the cytoplasm here, like this. Cytoplasm is present all over the cell. So this is the cytoplasm. Now when the cell is in a turgid position, a pressure is going to be exerted by the cell wall towards the cell contents. That is, the cell wall is going to exert a pressure on whom or on what? On inside of the cell, that is inside the cell contents of the shell. Let me just draw it. Draw the direction of the pressure. Here the cell wall is exerting pressure on insides of the cell. So this particular pressure is known as wall pressure. Direction of wall pressure is from the cell wall towards the contents of the cell. Let me just write it for you all. Pressure is exerted by the cell wall towards the contents of the shell, and another pressure is going to act in the same cell, and the direction of the pressure will be just opposite, that is in this direction. Now this pressure is known as turgor pressure. What is this pressure going to do? This pressure is exerted by the cell contents This pressure is going to be exerted by the cell contents in return of the wall pressure towards the cell wall. So both the pressure are sort of like equalizing each other. Wall pressure is happening, and in return of that, or in response to that, turgor pressure is happening. So both of them are happening simultaneously in a turgid cell. Let me just write that also for you all. What we can conclude is both, let's write it, both wall pressure and turgor pressure are equalizing each other. That is what we can conclude. Let us quickly have a look into the definition of each type of pressure, and then we can move on with our next topic. So wall pressure, the pressure which is exerted by the cell wall on the contents of the cell is called wall pressure. This was supposed to be off. Anyways, I'm sorry for the mistake. Okay, so the pressure exerted by the cell wall on the contents of the cell is called wall pressure. Since the cell wall is exerting pressure, that's why the name of the pressure is wall pressure. Cell wall here is exerting pressure. And just the opposite of wall pressure is turgid pressure, which is opposite of wall pressure. The pressure which is exerted by the cell contents in return on the cell wall is called turgid pressure. That was all about the two types of pressure that is observed in a turgid cell. So both of these pressures acting on the cell help the cell to maintain its turgidity, to maintain its turgid condition. Now let us move on with our next topic, that is flaccidity. And we are going to be understanding flaccidity with the help of this particular diagram. Flaccidity, we can say it is just the opposite of turgidity. You can see that this is a plant cell. It has this incredible cell wall which gives unique nature to the plant cell, and then this side is the cell membrane. And just the opposite of turgidity is happening. Simply what is happening? All the cell contents are shrinking. In which direction? Inside. Inside of the cell. The direction of shrinkage is in this direction. The whole cytoplasm has shrunken and the organelles as well. So all the inside parts of the cell has shrunk, and this condition of the cell is known as flaccidity. So flaccidity means cell shrinkage, and the condition of the cell in its shrinkage stage is said to be flaccid. Let us have a look into the definition. It is just the opposite of turgidity. It is a condition in which the cell contents shrink towards the inside and the cell becomes loose, a condition which is called flaccid. Simply, you all can understand, it is just the opposite of flaccidity. Sorry, it is just the opposite of turgidity. What is happening here? The cell is becoming loose. That is, the cell contents are shrinking towards the inside of the cell. And this shrinkage condition of the cell is called flaccid. So flaccidity is a condition of the cell in which the cell contents shrink towards the inside of the cell and the cell becomes loose and droopy. One more word you can add here, which is droopy. Let me just write it down for you all. Cell becomes one second, let me just change the ink color. Okay. Cell becomes soft, loose, and droopy. Sometimes you will notice that the leaves are droopy. In a plant, the leaves are droopy. Why? Because of flaccidity. Because all the water or all the contents of the shell have shrunk. That is why the leaves look droopy like that. So that is what is known as flaccidity. Very simple topic to understand. Extremely easy. It is just the opposite of turgidity. All you need to know is what is turgidity. If you know what is turgidity, you can very simply understand what is flaccidity. Now, there are two more phenomena that happen due to flaccidity and turgidity. What are those phenomena? Plasmolysis and deplasmolysis. The diagrams of both the processes and phenomena are given here. In plasmolysis, what is happening? You can see the arrows are coming out of the cell. That means these arrows are indicating H2O exit, that is exit of water. Let me just change the ink color here because the color is very boring for this ink. Okay, we can use red. We have no other color seriously. Anyways, so this arrow indicates the H2O exit, that is the exit of water, and this arrow here indicates the H2O entry. And then this is the vacuole. You all can see, you all by now you all know that what are the things present inside the cell when we are discussing about these kind of processes. We only need to care about cell wall, cell membrane, the vacuole. This is the vacuole. In case of deplasmolysis, the vacuole is tight. The vacuole is tight, stretched. Okay. In case of deplasmolysis, the nature of the vacuole is tight, stretched, enlarged, and baked. These are all the different attributes that you can name in this vacuole of the deplasmolysis. That means by the diagram we can understand the water is entering into the cell. That means this is the condition in which the cell is in turgid state. So we can say that deplasmolysis is nothing different than turgidity. It is very similar to turgidity. When the cell has absorbed a lot of water inside and it has stretched and swelled up, that position is known as deplasmolysis. Water enters into the cell, which means. And in case of plasmolysis, it is very similar to flaccidity. The cell contents have shrunken towards the inside, you can see, and water is coming out of the cell. That is, the cell is losing water. That is, exosmosis is happening. One more important point that you can note about these two important phenomena is here endosmosis is happening. Endosmosis is happening, and here exosmosis is happening. And here the nature of the vacuole is loose, soft, and shrunken. That is the characteristic of the vacuole in both the scenarios. Now let us quickly have a look into the definition. Plasmolysis, it is a condition in which the cytoplasm of the cell shrinks from the cell when the cell is placed in a hypertonic solution. The only difference between plasmolysis and flaccidity is the presence of hypertonic solution. If you remember, when we were discussing about turgidity and flaccidity, we said that these two things do not involve the presence of any type of solution, but plasmolysis involves the presence of hypertonic solution. So whenever the cell is placed in an hypertonic solution, at that time, what happens is the water comes out from the cell and the cell contents shrink. That position is known as plasmolysis. So when we were discussing about hypertonic solution, we could have discussed this when we were discussing about hypertonic solution, but I kept this thing separately for this portion of the chapter because it is much more easier to understand that way. Okay, so the only difference with plasmolysis is when the cell is placed in a hypertonic solution. It is a requirement for the process of plasmolysis to take place, or for the condition of plasmolysis to show up. That is all about plasmolysis definition. Now, deplasmolysis, just the opposite of plasmolysis, water enters the cell again. Now, if we have seen here, if we saw that plasmolysis involves the presence of hypertonic solution. That means if deplasmolysis is the opposite of plasmolysis, then it must be involving the presence of hypertonic solution, because isotonic solution cannot be the requirement of any of the processes, because in isotonic solution no change happens, it is just a neutral state. That's why we can say that this deplasmolysis condition requires hypertonic solution. So this is also a condition in which the water enters into the cell again when the cell is placed in a hypertonic solution. That is all about the two different phenomena of plasmolysis and deplasmolysis. Remember, this point, the presence of the solution, is very, very important when you answer your questions, because people and students get confused. The presence and the absence of hypertonic solution creates the difference between plasmolysis and flaccidity, and the presence of hypertonic solution creates the difference between deplasmolysis and turgidity. So make sure you keep that in mind. Now, our last topic for today will be significance of turgidity in plants. It's going to be a very small topic. We will just discuss it quickly, and by that we are going to finish today's video. The first point is turgidity provides rigidity to soft tissues like leaves. Soft tissues like leaves. You can see in this diagram, in the first diagram, the leaves or the cells of the leaves, this is just a cell examined in the leaf, and the leaf is in a turgid state. Sorry, the cell of the leaf is in a turgid state. So let me just write it for you all. This state of the cell, this is the leaf cell. This is a leaf cell, and it is present in a turgid condition. Hence, the plant is also very, very upright. That is, the plant looks fresh. You can see that the plant looks pretty fresh and alive. We can see and tell the plant has life in it. Plant looks fresh. The plant here looks fresh. But on the other hand, in the diagram beside this, we can see that the plant looks very droopy. Right? As if there is no life in the plant. It looks droopy. And it doesn't look fresh. It looks very dead and droopy and exhausted. This difference is seen because of the turgidity. In the first diagram, the cells are in a turgid condition, but in the second diagram, the cells are in a flaccid condition. That is why turgidity is very, very important for plant cells. This is flaccid condition. That's why the presence of turgidity in plant cell is very, very important to give them a tight, rigid structure and look, which will help them in looking tight and alive, which will help them in looking fresh. And in this case, the leaves actually have become very soft. We can also mention that point here. The leaves have become soft. And hence the leaves are looking this way. The second important significance of turgidity is turgor pressure helps the plants to push through the hard ground. Okay, let me just remove this ink for you all so that you can see this point. I'll just write it here. Turgid. These are the leaf cells. The second important significance of turgidity in plants is turgor pressure helps the plants to push through the hard ground. In many diagrams, in many places, you will notice that plants have come out from a wall or a cement. You can see hard concrete. In hard concrete also, many roots are visible. That stamina is provided by turgor pressure to the plants that push is provided by turgor pressure to the plant. Because of that strength, that power, they can grow in those areas as well. So that is a very, very important point for the survival of the plant. So this is important for the plant survival in harsh conditions. Plant survival in harsh conditions. The third important significance of turgidity is turgor in root cells develop root pressure. We have already discussed about root pressure in our previous sessions. Please go and check the videos out if you haven't seen them. Turgor pressure in the root cells help to develop root pressure. Root pressure is very, very important because it helps in the absorption of substances from the ground. So root pressure is important in plants because it helps in the absorption of substances. Helps in the process of absorption. Helps in the process of absorption in plants, and also helps in the upward movement of sap. We have also known about it. We have already discussed about all these things in our previous portions when we were learning about root pressure. And the last significance of the turgidity in plants is turgidity helps in the opening and closing of stomata. When we were discussing in a previous session about some importance of osmosis in plants and animals, then we got to know about a point that in plants, osmosis helps in the opening and closing of stomata. It is basically due to this condition which is known as turgidity, which we have discussed already in today's video. It's turgidity that helps in the opening and closing of stomata. What happens is sometimes during photosynthesis, there is a need, there is an extreme and continuous supply of water. There is a requirement of the continuous supply of water during photosynthesis. Therefore, what happens is the attraction of the cells, plant cells, for water molecules is extremely high. Hence, there is a constant pool of water molecules which is present during photosynthesis in the plant, and the cells are also in the turgid condition. So when the cells are in the turgid condition, wall pressure and root pressure exist, and due to the existence of wall pressure and root pressure, the stomata opens. This is a mechanism of stomata, guard cells and everything as a whole. We are going to be studying about this mechanism in details in the higher classes, but for the time being, all you need to know is wall pressure and turgor pressure develops, and hence the stoma opens. Stoma opens. Don't get confused with the term stoma. Stoma is nothing but the singular form of the word stomata. You can see single hole. So stoma means single hole. Stomata means multiple holes. Singular. And stomata is the plural form of the word stomata. That is all about the significance of turgidity in plants. And by that we have come to an end with today's chapter, today's portion. We have already discussed about everything that we needed to discuss. It was a very short video about today. If you have understood this video, then please share with all of your friends and help them as well. And definitely like and subscribe to my channel for getting more of such further updates. And if you have any doubt, then please do not hesitate to write in the comment box. I'll definitely answer them for you all. Till then, stay happy, stay safe, take care of yourselves. Peace.